Welcome back to Plus 2 Biology, the chapter Human Health and Diseases. Today we are moving to the second session of the chapter. It deals with immunity, vaccination, immunization, etc. So dear students, the major topics we brief today include immunity, innate immunity, acquired immunity, structure of antibody, active immunity, passive immunity, vaccination, immunization, allergies, autoimmunity and immune system of the body. Okay, at least some of these topics we can elaborate today. So, tell me what is meant by our immune system. You know we have digestive system, excretory system, you have studied all sort of systems in plus 1 and also the reproductive system you have learned this year. Then tell me what is meant by our immune system. Okay, which are the major parts of our immune system? Anyway, you know that our immune system confer what? Resistance to our body. Our body is protected from number of pathogens or attacking microbes by the action of our immune system, where components of our blood take part major role, you know. Okay, anyway, we have a subtopic immune system in detail. We discussed at that time what are the major organs and what are the major cells and parts of immune system. Anyway, so immune system is conferring our resistance. You know that. Then define immunity. What does immunity means? The overall ability of a host to fight against pathogens or disease-causing organisms is called immunity. And it is conferred by immune system. So, our ability to fight against infectious agents is known as immunity. Okay, so we can group the immunity into two main categories. The one is innate immunity. The second one is acquired immunity. From the word itself, you can get the meaning what is meant by innate immunity. Okay, just understand. Innate immunity is non-specific. Everyone possesses this irrespective of the pathogen. There is no any specific attack by this system. It will not screen a particular type of organisms. So innate immunity we can say this is non-specific immunity. Okay, it is not showing specificity to a particular type of organisms or like it is common for all. Okay, this immunity, innate immunity measures we can discuss under barriers. These are certain barriers in our body. The first one we can discuss as physical barriers. Physical barriers. You know, immediately what comes to your mind will be our skin. Our skin protects us from the external agents. To a certain extent, it is deposited with all sort of necessary things, you know. So the skin is acting as an important immunological barrier, which is a physical barrier. Okay, skin is an example of what? Physical barrier, which protects us from various germs, and the mucus coating. Mucus coating present in the inner lining. Outside, okay, we are protected by our skin, and any germ when it enters to our body through the nasal, okay, oral or whatever passage, or how we protect it? It is through the mucus lining. So the physical barriers, inside is mucus, outside it is mucus lining, and outside it is skin. These are the physical barriers. Okay. Then when we discuss about the next type of barriers, we call it as physiological barrier. Okay. Physiological barriers. I mean, with the help of various physiological activities, we are producing certain, by various metabolism, we are producing certain chemicals, and this gives us immunity. As a physiological barrier, we can just take the example of HCl in our stomach. Okay, and this pH is attacking or killing many germs which enter through our mouth. Okay, so HCl is a physiological barrier, HCl in the stomach. Then lysozyme. Lysozyme in tears. This is in the stomach, HCl in the stomach. Lysozyme in tears. Saliva in the mouth. These are examples of physiological barriers. By various metabolism we produce these chemicals. It gives us what? Immunity. So physiological barriers, example, the acid in the stomach, lysozyme in tears and saliva in mouth. Then the third type of barriers, cellular barriers. Certain cells give us immunity. Which are the cellular barriers? Immediately what comes to your mind is WBC. It is acting as an important cellular barrier. Okay, WBC. Okay, lymphocytes. Then there are macrophages in the tissue. Okay, monocytes. Then polymorphonuclear lymphocytes. Polymorphonuclear leukocytes. Natural killer cells. These are acting as cellular barriers. Certain cells which provide us immunity include WBCs, macrophages, monocytes, polymorphonuclear leukocytes, certain type of WBC, natural killer cells, etc. Then there are certain cytokine barriers. Some chemicals, some specialized chemicals act as barriers. Cytokine barriers. Example for cytokine barrier is interferon. Okay, you are familiar. Interferons are the chemicals produced by virus infected cells. Okay, so such cells produce this chemical means immediately it prevents the spreading of the virus to the next cells, neighboring cells to a certain extent. So interferons are the chemicals which help in restricting the spreading of virus in our body. So these are the common barriers which provide innate immunity. So once again, the innate immunity is non-specific immunity which every person has and by which we are born. Okay, and the major immunity factors include, we can call them as barriers. The major barriers or the factors which provide innate immunity are physical barriers like skin, mucus coating, physiological barriers like acid in the stomach, then tears, and then what, saliva, like. Then cellular barriers include, cells giving immunity include WBC, then polymorphonuclear lymphocytes like WBC, natural killer cells, monocytes, macrophages, etc. Some of these produce proteins to attack the germ. Some of them go and engulf the germs. For example, these macrophages go and engulf the pathogen as such. So the different types of action, either they produce toxins, otherwise they phagocytose the germs. Okay, then cytokine barriers are the barriers like interferons which help in preventing viral attack to next cells. So we have discussed what is the definition of immunity and what is innate immunity, which are the major barriers which provide innate immunity. And our next topic is acquired immunity. Okay, the name gives you the idea, this is what we acquire. Okay, if we acquire this means we should have some experience or exposure early. That means it is specific. Okay, it is specific to certain pathogen. So we can say acquired immunity is the acquired one. Okay, then so it is specific to a pathogen. It is specific to certain pathogen. Very important point, acquired immunity is specific. Tell me how we acquire immunity. You are very familiar about the case of chickenpox and some diseases where we get resistance due to our own exposure to the pathogen early. Okay, the same thing we are going to discuss here. In acquired immunity, a person gets resistance by himself. How is? He is acquiring the resistance due to his primary exposure to the pathogen. Due to the experience of an early exposure. That means when a pathogen attack our body, okay, a pathogen is first time attacking my body, then my response to that, as a response to that pathogen, in order to process that pathogen, my immune system works. Okay, and the immune system produces antibodies to fight against it. Okay, then this response, I have an exposure to the pathogen and I am experiencing, my body is building a resistance by producing certain antibodies. Okay, then this is a slow exposure. That means it takes time and to process. This is the first time. It takes time to process that pathogen and I will get the disease symptoms. Okay, so if the chickenpox virus attacked me for the first time, okay, and when it spread in my body, within that time my immune system tried to process it and it will be a slow response. This is called primary response. Okay, in the primary response, as a part of primary response to process the chickenpox virus, my body produced number of antibodies. Okay, and some of these antibodies will be left as memory cells in my blood. Then imagine the second time or third time, the next exposure, okay, of the pathogen to me. The next time the pathogen is attacking me. Then that time the memory cells will be showing an activated response to this pathogen and immediately process the pathogen without even producing the symptoms. That's why I am not getting the chickenpox for the second time. Okay, that means the primary response to a pathogen, which is a slow feedback, slow action. I am not saved from its symptoms, I get the disease. But as a result of that exposure, the primary response, I am getting a number of immune cells in my body. This will enable me to attack the pathogen for the second and subsequent encounter in a heightened way. Okay, this is what is called a secondary immune response. The secondary immune response is an activated response so that the person is not even getting the symptoms. So, I am not getting the chickenpox at all. That means the virus might have entered, I am not getting any of the symptoms because my immune system, my body processed it in a high speed. So, the primary immune responses are always slow, whereas secondary immune responses are always heightened. The secondary immune responses are also called anamnestic response. Okay, so actually the acquired immunity is due to a primary response. Okay, and the response is slow one, whereas it is helping us to produce an acquired immunity where the secondary response will be a heightened response. It is also called anamnestic response. Okay. The highly activated response, secondary response is called anamnestic response. Both this primary and secondary responses are due to our two major immune cells. Okay, the primary response and secondary response are achieved by B cells and T cells. Do you know what are B cells and T cells? Both are lymphocytes. We can say B lymphocytes and T lymphocytes. What is the difference? You have learned already. B lymphocytes and T lymphocytes. Both are WBC, two types of WBCs. And the B lymphocytes are formed and matured in bone marrow. Okay, they are formed in bone marrow and matured in bone marrow. That's why B cells. Whereas T lymphocytes are formed in bone marrow but matured in thymus gland. That's why the name. Okay, origin is same but maturation takes place in the thymus gland for T lymphocytes. Anyway, both B lymphocytes and T lymphocytes are two types of WBC lymphocytes. The B lymphocytes both are formed in bone marrow, whereas B lymphocytes mature there itself. T lymphocytes are matured in thymus gland. That's why the name. Okay, another important difference, it is a B lymphocyte which is producing antibodies. B lymphocyte produces antibodies, whereas T lymphocytes are not producing antibodies. Okay, then B lymphocytes, therefore we can say any pathogen enter our blood, B lymphocytes attack them by releasing antibodies. So we can call they are doing antibody mediated immunity. B lymphocytes are performing antibody mediated immunity. So the B cells formed and matured in bone marrow, whenever they need to process a pathogen, they are releasing antibodies to process them. So their action is antibody mediated immunity. Whereas the T lymphocytes, okay, they are formed in bone marrow They are formed in bone marrow, matured in thymus gland. They are not producing antibodies. Whenever they need to process a pathogen, they are performing cell-mediated immunity. What do you mean by this? Lymphocyte, okay, will engulf a germ. If there is a foreign germ there, what the lymphocytes do? This lymphocyte go and engulf it and process inside. That's what is cell-mediated immunity. Okay, this is not releasing any protein or antibodies to attack. Instead, it phagocytoses the germ and process inside. That is what the cell-mediated immunity is. Anyway, we have discussed acquired immunity is the one which we acquire. It is specific and it is achieved by B cells and T cells. And B cells show antibody-mediated immunity AMI and T cells show CMI. I think these parts are clear. Again, we are going to discuss about the structure of antibody. That means that B cells fight against infectious agents by producing what? Certain specific defense proteins called antibodies. You are very much familiar about antibodies. In the lower classes onwards, you are hearing this antibodies like. So it is very familiar word, but today you are going to study the structure of the antibody and some of the details of that. Okay, so here is the structure of an antibody. You can just see it is having a fork-like structure, a Y-shaped structure, and you know it's a protein. Okay, it is made up of 4 polypeptide chains. See, here you can see 4 polypeptide chains are joined by disulfide bonds. Okay, and in the chains, if you listen properly, you can see 2 long chains and 2 short chains. Okay, the 2 long chains, the long peptide chains are called heavy chains and the 2 short chains are called light chains. The structure of the antibody we used to tell it is H2L2. Heavy chains 2, light chains 2. Okay, when I draw the structure of an antibody, you can just understand it properly. This diagram you have to learn for your exam point of view. So just practice with me how to draw the structure of an antibody. Okay, the antibody is also known as immunoglobulin. Another name of the antibody is it is an immunoglobulin molecule. Immunity providing protein, immunoglobulin. Okay, then there are many types of immunoglobulins like immunoglobulin A, immunoglobulin M, everywhere, right? Immunoglobulin A, immunoglobulin M, immunoglobulin E, IG, G. Just remember with the word AMEG, the name AMEG. Okay, AMEG. Okay, 4 major types of immunoglobulin molecules in our body. Then when we discuss the structure of an antibody or immunoglobulin, and as you have seen, there are 2 heavy chains and 2 light chains. Always keep in mind the heavy chains are in the central portion. Okay, the long chains. The light chains are always outside. Okay, this is the structural representation. This is simple representation. 2 heavy chains inside, long chains inside, 2 small chains outside. These chains are connected by disulfide bonds. Here is a disulfide bond connection. Here is a disulfide bond connection. Here is a disulfide bond connection. Okay. So here I have drawn 2 long chains, long polypeptide chains in detail. Okay, elaborated way. Then the 2 short chains. Okay, parallel to this, the 2 short chains. When you draw it with the scale and pencil. Okay, the 2 short chains. Then how are they connected? These 2 are connected by disulfide bonds here. And here there is a disulfide bond, as we have marked already. Here there is a disulfide bond between the chains. Okay, within the chain also disulfide bonds are there. Here there are disulfide bonds. This is a within the chain bond. Okay, when you draw, take care of this. Here also outside there is disulfide linkages like this. Okay. connect this. Very important thing what to label here in the diagram is, these are the disulfide bondings. Always keep in mind these three interpeptide. Here is the connection. Here is the disulfide bond. These two heavy chains are joined by this disulfide bond. Always keep in mind. Okay, the structure of antibody. And very important thing, here in the diagram you have to draw some sites where the antigen bind. Proper clipping space, like you have drawn enzyme substrate complex site. Any special way, just the antigen binding site is this site. They should have some active sites for binding with the antigen. Okay, so we can, when we label these things, this is the antigen binding site. Okay, antigen binding site of the antibody. Antigen binding site. Okay, this side and this side. With these two arms, it binds to the antigen. So this is the antigen binding site. This is next one. Antigen binding site. If this is an antigen, it will be binding the antigen like this. Okay. And if this is the antigen, when we compare the size of the antigen and antibody will be very small. Okay. Number of antibodies are required to process one antigen. So antibodies bind to the antigen with the help of this antigen binding sites. Then you have to strictly label this as the light chain, and this as the heavy chain. Okay, light chain, heavy chain. So when we discuss the structure of antibody or immunoglobulin, we tell it is H2L2. Its structure represents H2L2, means 2 heavy chains and 2 light chains. Draw and study the structure of antibody. The same thing can be asked as the structure of immunoglobulin. So, you know, antibodies are produced by B lymphocytes, process the pathogen, and you already understood in order to process the antigen, antibodies are released and the antibodies kill the pathogen. And how the cell mediated immunity works? The cell as such phagocytose. Okay, the cell as such process the pathogen. Okay, cell mediated immunity is having an important role in graft rejection. Make it very clear. Graft rejection is a function of CMI. That means grafting means what? In plants you are familiar, similar name is used in animals also. In human, grafting means any body organ transplantation. Whenever we receive, we need to receive a foreign transplant to our body, and it is considered as a foreign substance, and the rejection is done by cell mediated immunity. The cells are fighting against it. We have to do so many procedures to suppress it. Okay, immunosuppressor should be used. Anyway, cell mediated immunity, keep in mind, it is responsible for the graft rejection. Okay, it's not producing any antibodies. The cells are such function to process the antigen or foreign substances. Okay, and by this we stop for today, and the remaining portions we discuss in the next class one by one. So we have discussed what is immunity, the definition, then innate immunity and its barriers, then acquired immunity by the B and the T cells, antibody mediated immunity and cell mediated immunity, and we have also completed the structure of antibody, draw and study, and always it is mistaken by students drawing the ends left and right. Okay, drawing the ends inside and outside are wrong. Okay, anyway, the active and passive immunity and the remaining topics we discuss in the next class. So, see you in the live session. Thank you.